Hello, thank you for joining us today for a webinar titled Acid versus Acidified Foods, looking at the microbiological safety, spoilage, and regulatory considerations. So this webinar will feature presenters Martha Kimber of Eurofins Microbiology and Dr. Ashwini Bag of the Clorox Services Company. So our presenters have prepared an in-depth overview on acid and acidified food products, their classifications, their unique product characteristics, and how they impact and can comply with regulatory considerations, microbial safety, and quality standards. As we get started, I'd like to read a brief background on each of our presenters for context of the presentation. So first we have Martha Kimber. Martha serves as Senior Technical Manager in the Advanced Food Microbiology team at the Eurofins Micro Lab in Fresno, California. She has been part of this team for over 14 years and specializes in providing clients with process authority support and customized challenge studies for a variety of food products based on their regulatory requirements and research objectives. So prior to joining Eurofins, Martha worked with Hain Celestial, the California Department of Public Health and Food and Drug Lab, and Hall Wines. Martha holds a bachelor's degree in microbiology and a master's in food science from UC Davis. Our other presenter today, Dr. Ashwini Bag, is a senior scientist at the Advanced Measurement Sciences team at the Clorox Services Company in Pleasanton, California. She has been a part of Clorox for almost 11 years in the capacity of a product developer and microbiologist, and in her current role helps the foods and cosmetics R&D teams work on microbiology risk assessments to support multiple product launches to market every year. Prior to joining Clorox, Ashwini worked at Mead Johnson Nutrition. Ashwini holds a PhD in food science and a master's degree in biochemistry from Utah State University. All right, well, with that introduction, I'll now hand it over to Dr. Bag to begin. So Ashwini, please go ahead.
Hello, everyone. We'll start with the webinar, and the agenda is as listed on the slide here. We'll talk about considerations for product formulations, microbiological safety and spoilage concerns, spoilage organism challenge studies, regulatory requirements, and then classification of acid versus acidified foods, and also talk about some of the processing methods, challenge studies, and finally, FDA filing process for acidified foods. So let's talk about considerations for product formulation. A product developer formulates for consumer delight and consumer safety. The factors that impact microbial growth in food products are generally categorized into intrinsic and extrinsic factors as they pertain to the formula. Some of the properties in perspective of acid and acidified foods that impact the growth and survival of microbes in formulations are pH, type of acidulant used in the formula, preservatives, water activity of the formula, and storage conditions. So let's look at how pH impacts formulations. pH is referred to as the negative log of hydrogen ion concentration. Microbes grow over a wide range of pH. Ensuring that we stay below a pH of 4.6 is per 21 CFR 114, which ensures that Clostridium botulinum spores do not germinate in the finished product. Use of a combination of organic and inorganic acids may be useful for formulation, where one of the acid aids with lowering the pH and the other in its protonated form ensures that the product is protected. The protonated form of an organic acid is effective in causing a microbial kill. A product with particulates may take longer to equilibrate to a designated pH, depending on the number and the distribution of those particulates in the product. So now let's talk more about the acidulant types. Organic acids are very effective as acidulants and do not allow for the growth of microbes when the product is formulated below their pKa, since they exist in protonated or undissociated state. In a study, when the broth was adjusted to a pH of 4.0 to 5.5 with organic acids, the order of effectiveness to prevent the growth of E. coli O157H7, which is a pathogen of concern, was in the order of acetic acid being the most effective and tartaric acid being the least effective. From a formulation perspective, if you look at all these different acids listed here, lactic acid has strong dairy notes, while acetic acid or vinegar has very strong sour notes. Therefore, the use of acid in a product is most likely determined by its organoleptic properties, its pKa, and the pH of the finished product. In another study published by Bright et al., in a brine cucumber matrix at a pH of 3.5 with 0.1% benzoic acid, 2.5% acetic acid required about 4 days to reach 5 log reduction of E. coli O157H7, whereas it took about 14.5 days to reach that same 5 log reduction in citric acid buffer. Another factor to consider would be the water activity. So water activity, which is referred to as AW here, is the ratio between the vapor pressure of food itself when in a completely undisturbed balance with the surrounding air media and the vapor pressure of distilled water under identical conditions. In a formulation, it is important to know what type of microbes might grow and also know the minimum water activity that supports this microbial growth, as shown in the table on the bottom right-hand side of the slide. Lowering the water activity can prevent microgrowth, but does not necessarily cause microbial die-off. Some of the formulation factors which control for water activity in a formulation would be the concentration of salt in your formula, the concentration of sugar in the formula. Additionally, any water binding agents which are used in the formula will also control for water activity. Some processing steps such as drying or heating, if used in the formula, would also control for a reduction in the water activity, which can be verified by measuring the water activity before and after the processing. Packaging and storage temperature of the product would also have an impact on water activity. Another important factor which controls for the presence of microbes in the formula is preservatives. The use of preservatives, both chemical or natural, in a formula controls the population of microbes in a product, and it can help extend the shelf life of your product. The preservative efficacy may depend upon the product pH. You want to choose a preservative for your product that is cohesive to your processing and doesn't degrade upon processing. If you have data showing it degrades, you may need to add some overages of this preservative if it's still deemed useful despite the processing and pH of the formulation. If your finished product is an emulsion, it may be important to know about partition coefficient of the preservative, if any. Similarly, propensity to solubility in a specific phase is also noteworthy. The mechanism of action of preservative and knowing the regulatory landscape of preservatives is important. Some of the preservatives shown here are sodium benzoate, potassium sorbate, which are examples of chemical preservative, and some of the natural preservatives would be rosemary oil, etc. Preservative suppliers have experience and expertise to help guide formulators with levels of stability and log reductions in a specific formula. Additionally, such studies are also conducted in broths. Additional studies can be conducted in the formulation of your choice to verify these claims. So now let's talk about the microbiological safety and spoilage conditions. Acidified foods have a very strong safety record due to extensive US regulations which contribute to the safety of acidified foods. There are few instances of foodborne illness outbreaks from acidified foods, which are most likely linked to improperly acidified home canned products. The pathogens of concern most likely would be Clostridium botulinum, Clostridium perfringens, Staphylococcus aureus, and Bacillus cereus, which need favorable conditions to grow and/or produce toxins. Clostridium botulinum is inhibited by pH level of 4.6, making it less of a concern for properly acidified foods, as we'll learn in some of the future presentation slides. E. coli, O157H7, Salmonella, and Listeria are highly pathogenic even at low levels. So these pathogens can survive in unfavorable or acidic environments, posing a public health risk. They are primary organisms of concern for acidified food products. Some other concerns in the food products would be microbial spoilage organisms. So, for example, it would include microbes like yeast, molds, lactic acid bacteria, and acid-tolerant bacilli, which do not pose a food safety risk, but they can still cause spoilage and economic loss. Some spoilage organisms are heat resistant and can survive even pasteurization treatments. Some examples of these would be heat resistant molds and Alicyclobacillus species. And control measures for such spoilage microbes would include using high quality ingredients, preservatives, and strict adherence to good manufacturing practices and sanitation controls. Let's talk about spoilage organism challenge studies. Spoilage organism challenge study considerations would include manufacturing location control, because these are usually non-sterile environments, using good manufacturing practices, standard operating procedures for sanitations and environmental monitoring programs in manufacturing facilities. The nutritional makeup of your product based on literature review and monitoring the manufacturing environment would help you understand which specific microflora can propagate in your product. And different production locations may have different microflora present. So if you have multiple manufacturing locations, it might be important to understand what type of spoilage might occur based on these different locations. The challenge test is designed to determine how the least robust form of your product will perform if it encounters certain logs of quality defect causing microbes. And a way to understand this would be to take the product and inoculate it with known concentration of spoilage microbes after storing it at a given temperature and testing it at regular intervals. The objective of these challenge studies would be to gauge products' ability to counter spoilage organisms under specific storage conditions. It's important to understand what the performance criteria for your specific product is For example, it may be less than 3 logs of growth of lactic acid bacteria in high acid products. Here is an example of a published spoilage organism challenge study. Consumption of spicy foods and hot sauces is currently a trend worldwide, and this is a spoilage test conducted in a cold-filled whole process. The cold-fill hot sauce spoilage challenge test for the effectiveness of acetic acid versus citric acid as levers to reduce population of spoilage organisms in hot sauce at 3 different pH levels. They are either a pH of 3.2, 3.5, or 3.9. The left panel indicates for presence for a spoilage challenge study conducted on lactobacilli, and the right panel indicates for the presence of yeast. The performance criteria as deemed right by this manufacturer was a 5-log reduction of spoilage microbes, which may or may not be a typical criteria used in the industry. In left panel, C is not a desirable result. And in the right panel, A with citric acid, B with citric acid, and C with acetic and citric acid is not a desirable result, as we see an increase in the number of total microbes post-inoculation. An alternate processing parameter or a preservative can be used if we find an undesirable result in our challenge testing. To conclude, acetic acid is more effective at controlling spoilage at a pH of 3.2 and 3.5. That leads us into regulatory requirements.
Thank you, Ashwini. So now I'm going to talk about the regulatory requirements, which are not at all confusing, as you will see. And yeah, just a quick summary. We are talking about the US FDA's 21 CFR 114 for acidified foods, and that defines what an acidified food is, requirements for processing and record keeping. Also 108.25, which is emergency permit control, so the requirements for the permits, and also 117, which is kind of the umbrella catch-all for food and beverage, which includes good manufacturing practices, hazard analysis, and risk-based preventive controls for human food. Also know what the requirements are within your state, but I'm going to speak to California because that's where Ashwini and I reside. And here in California, you're subject to the cannery inspection program, and you need to submit a request for pH form. And in California, the state acts as your processing authority, and they will issue you an S letter, which is the same as a process authority letter. All right, and here's the legal definition of acidified foods from the reg. So acid foods mean foods that have a natural pH of 4.6 or below. For example, canned tomatoes. Acidified foods means that you have low acid foods to which acid or acid foods are added. And in general, these have a water activity greater than 0.85 and a finished equilibrium pH of 4.6 or below. Often these are called pickles or pickled thingamajobs, whatever they are. There's also exemptions that are listed, so carbonated beverages, jams, jellies, preserves, acid foods that contain small amounts of low acid foods and have a resultant finished equilibrium pH that does not significantly differ from that of the predominant acid or acid food. Also, foods that are refrigerated are excluded from coverage in this part. So I had to read all that because we're going to go into a lot more detail for some of this wording here. Also, there's a thermal processing requirement under 114.80. Acidified foods shall be thermally processed to destroy vegetative pathogens and spoilage organisms. You are permitted to use preservatives that inhibit reproduction of microorganisms of non-health significance, which means spoilage organisms, in lieu of thermal processing. So basically, they're saying you're required to thermally process, but you do have an out if you are formulating your product to inhibit spoilage. To make this a little bit trickier, back in September 2010, there was an issuance of the FDA's draft guidance for the industry, acidified foods. And this was a very prescriptive document that kind of described everything about acidified foods and how to classify them. In this guidance document, they considered water as a low acid food or low acid ingredient, which is a big deal, as you'll see in the coming slides. Also, they defined what a small amount was, and also a pH shift, what kind of pH shift after adding your acidulant was allowable. So again, very prescriptive. However, in 2015, this draft guidance document was rescinded, and that brings us to present. It should not be used anymore as a guidance document, but occasionally I have seen, and FDA has seen, that some inspectors are still using it. Just keep an eye out. You're not subject to the draft guidance anymore, and it's improper to still be using this document. Another update regarding acidified foods occurred at the IFP 2022 roundtable session. So this was a big conference in Pittsburgh, and we were discussing acidified foods. And Glenn Black of the FDA, he spoke, and he's a big deal for the LACF group, which governs the filed LACF and acidified foods. And he said that there will be no guidance document issued for acidified foods. So that's been withdrawn, and they're not going to make another one. FDA will not consider water as a low acid ingredient. So this is clearly stated. They're no longer going to consider it a low acid ingredient, which will be a big deal coming up for formulated acid versus acidified foods, as you'll see. Glenn Black also said that for products already filed, if you reach out to the LACF group, you can request clarification, and they can also assist with the classification if you like. He said that preservatives are no longer restricted to only sodium benzoate and sorbate, so other preservatives are allowed. And aside from water-based beverages, really, in my opinion, there weren't really any other significant changes to the policy. He did say that a 5-log reduction of vegetative pathogens is not an absolute requirement, but compelling data must be submitted for a lower reduction. And the way I interpreted this was that they're looking for something similar to what the Almond Board of California did, where they argued from a 5-log reduction down to a 4-log reduction based on prevalence data that was taken over several years. It's a huge amount of data, quite a bit of work. So it's possible they're not shutting the door completely, but you have to do a lot of work to achieve that. All right, so this is where we stand currently as of 2025. Acidified foods are shelf stable. They contain low acid components plus an acidulant or acid food. They have an equilibrium pH of 4.6 or below and a water activity greater than 0.85. They shall be thermally processed, and the process must address the container closure. They must be filed with the FDA using forms 2541 and 2541E. However, there's still an out. If you want to cold fill, you can do so if you have supporting scientific data. And overall, these products are covered by the requirements of the FSMA PC rule, which is 21 CFR 117. So Another summarization of all that is that products that are not considered to be acidified foods are refrigerated or frozen, carbonated, fermented, naturally acidic foods, foods with a water activity that is below 8.5, or formulated acid foods. I would still stress that you need to watch out if you're adding large amounts of low acid ingredients to any of these, because that can change the game a little bit. Also, alcoholic beverages are covered by the TTB, whereas these listed on the slide are covered by 21 CFR 117. All right, so now I'm going to talk about the classification of acid versus acidified foods and the things that I think about as I'm looking through these formulations. So big picture, these regulations are designed to prevent growth of Clostridium botulinum. Produces a very serious toxin. We don't want that, of course. And the preamble to the acidified rule regulation is focused on products that contain a large percentage of non-acid ingredients, say a cucumber, or large particles of non-acid ingredients, and thinking about how long it takes for them to reach a pH equilibrium below 4.6. And they use the wording may be called or purport to be pickles. So again, this would be chunks of low acid ingredients like a cucumber or an onion or a clove of garlic. And if you think about it, something that has a very large particle, it's going to take longer for the acid to fully penetrate to the center of the particle. Whereas if it's a very fine particle, then it's going to equilibrate much more quickly. And then here's a quick recap again, because the regulation does get a little bit tricky. So again, acid has a natural pH below 4.6, shelf stable, contains small amount of low acid ingredients, and this less than 10% small amount is no longer accepted by FDA as a formal criterion for small amount, since that draft guidance document was rescinded. In terms of acidified foods, it has a large amount of low acid ingredients, and there's a pH shift after adding a acidulant, and acceptable pH shift is also no longer defined after that document has been rescinded. So what do you do with all this? One option is that you can work with an experienced process authority to classify your product. There's no assurance that the FDA will agree in every case, but it has helped in the past. If you're classifying the product as a formulated acid food, make sure that your letter has scientific justification for how you came to that decision. And if it's classified as an acidified food and the food is cold filled, non-aseptically, or if it's not thermally processed at all, make sure that you work with your PA to design and execute challenge studies, or you have, again, scientific data backing up what you're doing. There's also an option to do a voluntary filing under FDA Form 2541E. I haven't done this too often, but there is an option on the filing website. All right, and part of this classification, a big part of this is that you need to test pH of all of the components, and there are very specific instructions for the methodology of how to test pH, and that's in 114.90 sections A through B. So they describe exactly how to do it, if you have chunks in there, if you're trying to reach equilibrium, how to standardize your pH meter, all of that. And the way that I think about it as I'm going through a classification is I start step one looking at the formulation itself, so the percentage of each ingredient by weight, as well as the pH of each ingredient. Then we look for step two at the acid ingredients plus the water that is used in the formulation, and we measure the pH of this acid-water mixture. And then for step three, we grind up any solids or puree, we add it to all the remaining low acid ingredients from step 2, and then we determine the maximum equilibrium pH, so the pH of the total formulation. So basically, the goal of this part is we want to look at the pH of the acid plus water component and the pH of the finished product to see if there's a large shift there. Other considerations that are important to acid versus acidified foods. So several folks that used to be in our team, they worked on a really great document that was published in Food Protection Trends. And it's more of a qualitative evaluation to assist in the classification. They have charts and reasoning behind why they make certain decisions. It's very helpful. I don't agree with all of it there, but I think that there's a lot of good information there that you can cite. I would be remiss if I did not mention the great Fred Bright in talking about acidified foods. And recently he's been working on modeling tools for looking at the buffering capacity of different food ingredients and basically looking at the buffer ratios among the ingredients to see how the pH will change when you're mixing the ingredients together. Ideally, in the future, hopefully we can use a buffering capacity number to have a demarcation between acid and acidified foods. I don't know if it's there yet, and I haven't seen regulatory bodies approving of this method, but I do think that this is a tool that we should keep working on, we should keep looking into, and hopefully we'll get there. Because if your product doesn't have a lot of buffering and it instantaneously reaches the pH that you're going for, then where's the safety risk? So big picture, I really like what he's doing, and I hope to see this move along. All right, to talk about some processing methods for these products. First off, you shall do thermal processing, again from the reg, and the goal is to inactivate the vegetative pathogens and the spoilage organisms to make sure that your product's shelf stable. And some examples that pasteurize the product would be a kettle cook with an ambient fill or HTST with ambient fill, and that's going to treat the product only. There's also examples that treat both the product and the package, and again, there's a requirement in the reg that says that you treat the product and the package, so this is important. And that would be processes that either occur in the container, like a water bath or tunnel pasteurization, or perhaps a hot fill hold with tipping or inversion, or HTST plus aseptic. So those are all different options that treat both the product and package together. Some thermal processes to think about. So they should be designed based on the most heat-resistant organism that's present in the food. And so, of course, for low-acid canned foods, usually you're retorting for Clostridium botulinum, for example. However, for acid and acidified foods, your targets are a little bit different because you're not as worried about Clostridium botulinum. It can't grow at this low pH. Rather, you're at a pH between, 4-ish to 6. You're more worried about spores of acid-tolerant spoilage organisms like Bacillus coagulans, Licheniformis, and Clostridium pasturanium. And when you get to the lower pHs, so below pH of 4, you're more worried about yeast ascospores, molds, non-spore forming bacteria like acetic acid bacteria or lactics. And below are several references. This Flug book, it lives on my desk, so that's a photo from a book on my desk. And then Fred Bright, again, who is the guru for all things acidified. He has really great documents that you can reference with times and temperatures to address vegetative pathogens. And that would be these two thermal processing documents on the right. All right, so another thing that you'll see if you're looking through the reg is the option to cold fill hold. And this implies that it's a refrigerated food, but actually it's not. This refers to cooling after a thermal process or just filling at ambient. So usually these are more ambient rather than cold processes. And in this case, a formulation control is used which utilizes the product's intrinsic parameters. So the items that Ashwini was talking about earlier, like pH, acidulant, preservative, and you're using the product's formulation itself to achieve microbial lethality against vegetative pathogens. So again, you need supporting literature, and there's a lot of great literature by Fred Bright and others. Or you can generate your own scientific data from an inoculated challenge study to demonstrate that your product formulation can achieve that die-off of pathogens. The product pH, acidulant, and any preservatives that are added to the product may be used as effective microbiological controls. And these formulation factors are going to become critical factors to your product. And now I'm quickly going to talk about some pathogen challenge studies that we can do. All right, so again, this experimental design must be scientifically sound. We always refer to the National Advisory Committee for Microbiological Criteria for Foods. They have a wonderful document called Parameters for Determining Inoculated Pack Challenge Study Protocols. That kind of has a list of all the things you need to think through when you're designing a study appropriately. Also, Fred Bright, he has a great paper that he published regarding how to design these studies specifically for cold fill hold acidified food products. And then something to think about, if you have lots and lots of acidified foods, it's often impractical to test every product or formulation. So something that you can do would be to group similar products and then choose representative products for your study. But it's really important that you choose a worst case scenario and you're really carefully selecting what you're choosing. So you need to look at your formulation parameters, your pH, TA, salt, preservatives, and you need to make sure that they're at the worst case or a critical limit. And then you need to make sure that you're monitoring all of those parameters prior to and during the challenge study. Other considerations, so it's really important to consider what you're doing with the organisms that you're inoculating. So E. coli 0157H7, Listeria, and Salmonella are identified as the organisms of concern for acidified foods. Ideally, you want to use strains recovered from similar high acid products. Outbreaks in clinical isolates, so you want to have a mix. Usually we include 3 to 5 strains per cocktail. And each pathogen cocktail should be tested separately. So that would be 5 strains of E. coli in one cocktail, 5 strains of Listeria in a second separate cocktail, and etc. The culture should be acid adapted prior to inoculation to mimic real product conditions. And there's guidance from Fred Bright on how to do that appropriately. Another thing to think about is that you need to inoculate high enough so that you can observe the 5-log reduction, or you can demonstrate a 5-log reduction. So say, for example, your limit of detection by plating is 1 log CFU per gram. You want to inoculate at least 6 log CFU per gram so that if you have a 5-log reduction, you can still visualize that completely. And finally, when you are plating out or recovering the injured microbes, you want to be using a non-selective medium. If you were to plate injured salmonella on tryptic soy agar versus XLD, you're going to see a different result because it's going to recover more readily on the non-selective media as compared to a selective media. And then another thing to consider would be the incubation conditions. It's considered worst case scenario. to conduct the study at lower temperatures versus higher temperatures. And that's because these pathogens die off more quickly at higher temperatures and more slowly at lower temperatures. So if your production facility is located in a temperate area of the world or the US, and during winter it gets pretty cold and it's not a temperature controlled warehouse, they recommend using 10 degrees Celsius or 50 degrees Fahrenheit as your storage condition for the challenge study. Sometimes in climates that are very tropical and they never get that cold, we can conduct the challenge studies at a higher temperature. Another thing to think about would be the length of incubation. So the length of your challenge study. It should be aligned with the time that your product can be reasonably held at your facility before you release, because ideally you want to be able to hold your product under your control before it's released for distribution and you no longer have control over that product. So if you can't hold the product for, say, 3 weeks, you shouldn't have a challenge study that's 3 weeks or longer, because that data is not going to be useful to you in the long run. Here's a couple just sort of basic examples of pathogen challenge studies. So on the left is a condiment that has a pH of 4.4. It has things like soy sauce, onion, garlic, spices, a little bit of phosphoric acid. It also has some preservatives, sorbate and benzoate. We inoculated pretty high, so around 8 logs, and we observed 5 log reductions within 6 days. So yeah, pretty good hold time there, despite the fact that the pH was pretty high. And then on the right, we have a dairy-based sauce that has a pH of 4. And this product had a dairy component, a lot of vinegar in it, oil, spices, and sodium benzoate as a preservative. And we observed a complete die-off of all 3 organisms within 3 days. So for these 2 examples, we have minimum storage time, which for the product on the right would be 3 days at 10 degrees Celsius or higher. And those would be your critical factors for your process for your cold fill hold. And you can use the challenge study report as a supporting document in your FDA filing. All right, now going on to talk about FDA filing process for acidified foods. So the first thing you want to do is go and open an online account with the FDA, and you want to register your processing facility as a food canning establishment. Then you want to fill out your Form 2451E, which is your process filing details for the acidified food. As part of that filing, you want to include your process authority letter, which has all the details of your critical factors, whether it's your thermal process, your hold time, your product pH, all of those details. You would also want to include your challenge study, your pathogen challenge study final report, if you're using it in support of a cold fill hold product. The report should clearly state all of the product intrinsic parameters, so the pH, preservatives, etc., as well as the storage time and temperature required to achieve those 5 log reductions. And then finally, this was not really an FDA filing, but you also need to make sure that you're following the state requirements. So as I mentioned earlier, in California, the state acts as a process authority, so you want to make sure that you fulfill all of those additional requirements. And here are some links and further information as resources. So I linked to the actual regulation for 114, also the CDPH's cannery inspection and licensing program. And I'm a UC Davis alum, so I had to give them a shout out. They have really great resources for acidified foods, as well as better process control schools. They have some links to better process control schools specifically for acidified foods, which I find really helpful as well. So definitely check out all these links if you have more questions. All right, I'm going to turn it over to Scott now, but thank you so much for attending this webinar. We appreciate it.
All right. Thank you so much, Martha, Ashwini. So I'm going to go through top to bottom, maybe. There's a lot of, maybe clarifications on certain products. But first off, I want to ask, which ingredients typically contribute to buffering the pH? And maybe I'll combine that with another question. Where does liquid sugar fit into all this? Is there any considerations or special concerns there?
Ashwini, do you have comments about buffering? I'll let you go first.
Yeah, so from a formulation perspective, you kind of have to look at your formula holistically and figure out what the ingredients in your formulation are. I'll talk about maybe one ingredient. So I know there is a possibility that monosodium glutamate can contribute to buffering. There is a possibility that if you have any salts in your formulation, they are contributing to buffering. So I think you would have to really look at your formulation and figure this out. And as far as liquid sugar is concerned, some of the spoilage microbes of concern, I would think, would be Zygosaccharomyces bailii. So you could think about how would you control for that in that specific ingredient as it pertains to your finished product formulation.
Yep, and if you're asking about liquid sugar in terms of acid versus acidified, I think that this goes back to thinking about what is the pH of this ingredient. Sometimes liquid sugars are below 4.6, sometimes they're above 4.6. I would take a close look at that. And then you also want to look at what is the pH shift when you combine the low acid ingredients and the high acid ingredients together. All of that together will help you determine if it's acid or acidified. Liquid sugar is a gray area, though. There's been a lot of consternation on that particular ingredient.
Right. Very good. Thank you for that. The next question I'm going to ask is maybe a clarification. So I had a couple of questions on this matter, so I'll bring it up. Where is it stated that water is no longer considered a low acid? Is that just it was stated in the draft guidance which was rescinded, or is there additional information that specifically states that it isn't now?
Water is not specifically called out in the reg. It's not written anywhere. That's why I said that Glenn Black said it at the IFP conference. He said, I've told my team, the LACF team knows about this. We're not going to be considering it a low acid food any longer, but it's not written anywhere in the reg, and they're not planning on giving another guidance document to clarify that point.
Okay, thank you. I'll ask the next question, another clarification, I guess, or maybe for your resources that we can maybe provide after the fact, but is there public access to that modeling tool that you mentioned, Martha, about the buffering capacity in food?
Yes, yeah, so I need to find the exact link, but yes, you can play with it.
Okay, great. Yeah, that's something that we can provide perhaps afterwards then. How about this question? So the question is regarding the term formulated acid food. Can you speak about whether that's a recognized term with the FDA, or if there's any unique considerations with that term or its application and how it fits into what we talked about today?
That's also a very gray area. You guys are hitting it hard today. Okay, so formulated acid food. That is also not stated specifically in the reg, that terminology. And it wasn't in the guidance document that I'm aware of. But I have seen filings with that terminology be accepted.
Okay. All right, we'll take that then. All right, I'm going to switch gears a little bit then from the clarification questions and the additional resources. And I'm going to ask a very specific question. I'm guessing, again, might be very product specific, but maybe we can generalize it some. So the question being, for an acidified food, pH 3.9, that is cold filled into packaging, then high pressure processed, what is the recommended target organism? And I guess I'm curious on, how you might look at this and what other considerations you might have in identifying a target organism overall.
Yep. So I think that the main thing to think about in terms of target organisms would be pH first, because you can eliminate organisms of concern the lower your pH is. At a pH of 3.9, you're going to be more concerned about things like yeast and mold and lactics, acetic acid bacteria, in terms of spoilage and making that shelf stable. In terms of pathogens, absolutely, I would still consider E. coli, Listeria, Salmonella as your vegetative pathogen targets. And there's a few papers about HPP and these vegetative pathogens and the efficacy of HPP to target them in the literature, which I can share.
Okay. Perfect, thank you. Let's go back to, say, formulation some. Question being, can you recommend an acidulant that can lower pH without impacting flavor too much, or considerations there?
One could consider using phosphoric acid, although it's so I think we would need to do some research on our own, but like food grade phosphoric acid would be available, so that can be used. It's a strong acid and it'll reduce the pH. It'll have more impact on the pH, but has a little bit of a puckering flavor, but that can be masked by other ingredients in the formulation.
Okay. I guess another question then too on spoilage organisms. Are there specific types of spoilage organisms that we see most often causing issues for these particular products that people should target or be aware of?
I would think lactics are definitely a big concern in general. So lactics and yeast and mold, and then try and get information from the environmental monitoring program of your facility, as well as understand your ingredients really well to figure out what microbes might be of concern from a quality standpoint in those raw materials, and then those microbes can be chosen.
Okay. Thank you very much for that. Let's see, a question coming in about coffee. And is there any unique considerations with coffee as an acidified food, or maybe how it's prepared, or pretty open-ended question, I know, but thoughts on?
Yeah, so in general, coffee has a pH above 4.6. So most cold brew, hot brew coffees, I would say, are more pH of 5.5-ish, so they're a lot, they're definitely above 4.6. So they would be exempted from the acidified foods regulation. However, you would need to consider, is this coffee, is it shelf stable? In that case, you would need to comply with 21 CFR 113, which has a very similar requirement where you need to file with the FDA You need to have a process that addresses, pertinent pathogens and spoilage organisms. Except in this case, you're thinking more about the spore formers like Clostridium botulinum because it has that higher pH. So yeah, I would say although it doesn't qualify as an acidified food, it definitely has similar considerations.
Let's see, another question on what is included, which I believe you addressed, Martha. I'd like to understand how fermented products fit into the landscape and what food safety requirements there are for refrigerated fermented products. I know you mentioned that is not within the classification for acidified foods. Any gray areas there to discuss?
Yeah, so first off, if this is a fermented product where it's fully fermented, and if it's refrigerated, and you're not adding a lot of low acid ingredients after the fermentation, there's always that caveat. Are you adding low acid ingredients after the fermentation's completed? That could make things different. If it's refrigerated, it's going to be covered by 117. So it doesn't need to be filed, but you still need to think about how is this product going to be protected from a safety and spoilage standpoint if I'm adding low acid ingredients to the formulation. Yeah, also another thing to consider is if it's fermented and let's say you thermally process and you kill off all of those fermentation organisms and make it shelf stable. That is going to change the game a little bit. It may be qualified again as an acidified food if you're killing off all those fermentation organisms and making it shelf stable in a hermetically sealed container. It changes the game a little bit. So yeah, I would say depending what you're doing downstream can definitely turn it back into a gray area.
It's fair. Seems to be one of the themes of today's webinar.
I know, clear as mud.
Yeah. All right. Well, with that, I think we should wrap it up here. If you have further questions, we'll have a brief survey here that will go out to all the attendees. So feel free to answer that. Have the opportunity to put questions in there as well. If we didn't get to your question, we'll certainly follow up with you afterwards too. So I guess with that, I want to thank you again, Martha and Ashwini. Really appreciate the conversation today.
Thank you. Have a great day.
Thank you. Thank you all for attending.